a very interesting topic about plant whole genome sequencing. In this occasion, we are honored to welcome our speakers, Dr. Annabel Wibley from Bragato Research Institute, New Zealand, Dr. Yusmiati Chen Liao from Bragato Research Institute, New Zealand, Dr. Ratih Damayanti S. Hood, MSE, Director of Scientific Collection Management BRIN, and our moderator for today, Dr. Rernat Zahra Noviana, MSE, researcher from Research Center for Applied Microbiology BRIN. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. I will start to count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat feature. Now we are going to start our discussion for today, which will be led by Dr. Rernat Zahra Noviana, MSE. To Dr. Zahra, time is yours.
Thank you so much, Ibu Andalusi atau Ibu Loviana for the introduction. I would like to extend a warm welcome to all of the participants for the third webinar of Sequencing Laboratory of BRIN with the topic of plant whole genome sequencing. So good morning all. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, first allow me to introduce myself. So I am Zahra Noviana from the Research Center for Applied Microbiology, and I'm also a member of the Sequencing Laboratory. So with the other members, we are contribute to the ongoing initiative in genomics. And also I'm honored to be your moderator for today's webinar. So today we will have 3 speakers lined up, and each will present about approximately 30 minutes. So our first speaker is Dr. Annabel Wibley from Bragato Research Institute, New Zealand. And then later, we will have also Dr. Chen Liao, also representing the Bragato Research Institute, New Zealand. And then at the end, we will have Ibu Ratih Damayanti, the Director of scientific collection management. So with this, I would like to introduce our first speaker. So Dr. Annabelle Wibley from Bragata Research Institute. So Dr. Wibley is a bioinformatician in the grapevine improvement team, where her primary focus involves analyzing genomic datasets to uncover valuable insights into grapevine biology. And with a strong foundation in genomics, Dr. Wibley also holds an honorary academic position at the University of Auckland. And I already saw that she already here with us. So Dr. Wibley, could you hear me?
Yes, do you hear me?
Yes. Okay, so now the floor is yours.
Thank you. I will just share my screen with you. Okay, do you see my slides?
Yes, it's there.
Excellent. Thank you, Zara, for that very kind introduction. It's a real pleasure to be with you here today and to share some of the work that we've been doing to generate very high quality assemblies of grapevine for our work. Because I'm the first speaker in the session, I just wanted to start by talking a little bit about the nanopore platform and why it's such a good fit for our research interests. So as I'm sure you're all aware, what happens is you have protein pores embedded in a membrane on a flow cell, and DNA or RNA feeds through those protein pores. And as it does so, there's a sequence-specific change in the current that happens, and this current change is recorded. It appears as these so-called squiggle plots that you see here, and with very clever base calling models, these get deconvoluted into sequence reads. It's a really wonderful offering from our perspective for a number of features. The first one being that read length, it's read length agnostic, as they say. So you sequence reads as long as you're putting into your libraries. You can sequence amplicons, but in the assembly space, what's really wonderful is that provided you can extract DNA that's tens or hundreds of kilobases in length, you can sequence those in their entirety. And that really simplifies the assembly process. It's now also highly accurate, and I'll go on to talk a little bit more about how that's had an impact on assemblies. And it's very scalable, and it's scalable both in terms of the instrumentation and the flow cells that you can use, but also because the sequencing happens in real time. And what that means is you can start and stop an experiment when you get to the amount of data that you want to be able to answer your question. You can call both the canonical bases and also modifications on those bases, so methylated DNA at C and A positions, and Chen will go on to talk about a lot of that later on in her talk. And there's some other really neat features of the platform, including that you can do in silico enrichment or depletion, and so-called adaptive sampling, and again, really focus your sequencing effort on what you're after. So I give you a little bit of background into my kind of nanopore experience. I started working not on plants, but in birds when I first came to New Zealand in 2019. And at the time I was working on a threatened New Zealand native bird, which is called the hihi or the stitch bird. And we had really strong constraints around what we could do with that sample. And most specifically, what we were not able to do was send a sample overseas for sequencing. And so we had to have in-house capability. And we also wanted to leverage the huge advantages in assembly that came with having long read technologies. And so for that reason, I started with a MinIRON starter pack and generated a lot of long read data for these assemblies of the male and the female. And then we took a lot of the learnings from that and also applied to another project in the lab, which again has been quite recently published, looking at the genome of the myna, which is an invasive bird here in New Zealand. And this is some nanopore kind of publicity really around their accuracy from around the time that we were doing the bird assembly work. And I basically came in sort of probably where the green curve is. And as the accuracy improved through changes in chemistry and through changes in base calling and changes in the flow cell, so the quality of our assembly kind of tracked upwards. And this plot ends at 2021, where your raw read accuracy starts to have a median of around Q20. Just a couple of kind of definitions around assemblies. So the species that we're working on are diploid. They have 2 chromosomal copies. And what we were commonly capturing a few years ago is what we call a collapsed haploid assembly. And that's where you've got one representation of the chromosomes within your reference. And it's probably a mix of the 2 parental copies, just because they're not being very well resolved or very well phased. And those assemblies were what we were using for the bird work that I described, and they're very, very useful for population projects. But as the accuracies continue to track up, we actually now can get at fully phased diploid assemblies. So whereas the error that you had in the earlier versions of the chemistry was actually quite helpful for collapsing to a single haploid assembly, now because we've got that increased recovery of the variation, we can use that to really carefully resolve chromosomes. And so this is, I guess, where we're at with accuracy at the moment, but it does continue to improve. And I'm aware that there are likely some further announcements to be made in this space after London Calling in May. But the Q20 kit, the LSK114 chemistry, is giving you a mean simplex accuracy of 20. And then you also have this variation called duplex sequencing, which is essentially where you sequence both strands of a molecule within your library. And you can use those 2 strands to correct each other, and so further increase the accuracy of your reads. And here we're getting well above Q30 actually for our duplex scores. And that means that the more accurate data that you're feeding into your assembler, the better your backbone is. And just under 2 weeks ago, this preprint arrived in bioRxiv, which talks about gapless assembly of complete human and plant genomes using only nanopore data. So no need for PacBio Hi-Fi, no need for Illumina. With just nanopore, you can get chromosomal assemblies. Interestingly, in this project, the plants that they used were tomato and maize, and both of those are very inbred lines. And so they were not having to worry about heterozygosity in this case. They used duplex to about 40x coverage, and also had a certain amount of ultra long, which is defined as nanopore reads that are over 100 kb in length. So 30x coverage tomato, 16x coverage for maize. And they recovered many of their chromosomes telomere to telomere. And those that weren't telomere to telomere were relatively easy to fill the remaining gaps. So what I want to talk to you about today is our work on doing similar kinds of things. So assembling plant genomes. We work on grapevine. I'll give you a little bit of an overview of our particular project. But importantly, grapevine is highly heterozygous. It's clonally propagated. And often those haplotypes have been separate for a very long time. And so there's extra challenges that come in. I also have a disclaimer, which is I have titled my talk ONT only. It's actually not strictly true. We do have Illumina data sets for a number of purposes, and they are still quite useful to us. But I would also say that it's not essential to have those data sets to achieve that chromosomally complete near gapless assembly. We've looked at variations of our data that just use nanopore, and we still get pretty impressive statistics out the other end. Before I forget, I'm just one part of quite a large team working on grapevine improvement in New Zealand. It's led by Dr. Daryl Lismore. Amy and Banu do a lot of the lab side, and Solomon manages the plants, and other members of the team are working on these and other related projects using very heavily the nanopore platform. So we're actually the only institute within New Zealand that has a full-size Promethean at our disposal, and we're using it pretty heavily. And Chen will talk later about her work specifically on the epigenetics. So Sauvignon Blanc is the variety that we're particularly interested in, and that's really driven by the fact that it is the major kind of component of our wine import or our wine export market. So almost 3/4 of the vines that are grown in New Zealand are Sauvignon Blanc, and it's an even bigger proportion of our export market offering. And we're working actually in the fairly early stages of a very large program that's funded by government and industry in order to improve the genetic stock of Sauvignon Blanc that we have available to us. And that's because essentially we have one commercial clone that is being grown across the country, And that lack of diversity is a real point of vulnerability, and we've seen that with other monoculture crops that we have in New Zealand. And so the idea is that we will generate a diversity population of Sauvignon Blanc that can act as a source of phenotypic novelty, and that's particularly important to have solutions for this in this changing climate. And I think along the way, we're also going to get some very interesting insights into grapevine biology. There are a number of strands to generating that diversity collection, but really the major one involves generating new clones using a process of somatic embryogenesis. So we take progenitor cells from a Sauvignon Blanc plant and culture them in vitro. And we do that in a way that exposes them to chemical mutagens and also to physical stresses. And the idea here is that we will activate endogenous transposons to move around the genome and generate mutations, and that we're doing that importantly in a way that is not considered genetic modification or gene editing. And essentially, these are things that are very much not allowed under current New Zealand laws. And so this is really, although it's a relatively untargeted approach to improvement, it's one that we can be acting on now. And so those soma clones are then regenerated as plants, and we will go on to characterize those both phenotypically and genotypically. And that's really the driver for our wanting to generate a very accurate, high quality reference genome, is that we will start to be looking for very specific changes within these diversity collection. And the better quality our genome is, the easier it is to pinpoint what novel changes have been introduced. A few features of grapevine. So it's diploid and it's quite small genome, only about 500 megabases. Those are both very good things in the context of plant genomes. And as I said before, it's clonally propagated. We can't do conventional plant breeding because although we can cross these plants, when we do cross them, because they are reassorting the genotypes, it's no longer considered Sauvignon Blanc. So we really need to be acting for our project within that original genotype. Some other features that are interesting that are not so relevant here, but it's also important to be aware that usually these plants are grafted, and so we've got another set of genotypes and interactions to consider. And also chimerism, so where you have different mutations in different cell layers is well known to operate in plants and could be a complicating factor. Before I get into our assembly, we do quite a lot of sequencing for other groups within New Zealand. And one of the questions that we get asked a lot in the context of assemblies is, how deeply do I need to sequence? And so I thought I would at least speak to that a little bit with some data that we've used where we've downsampled quite high coverage data sets and looked at the impact on a number of metrics for the completeness and contiguity of an assembly. So just to orient you a little bit with these plots, what you have is decreasing coverage from left to right along the x-axis. And the plot on the left shows completeness. So, sorry. So what I want to show here really is that even at quite low levels of coverage, so we're talking about 12x coverage, so 6x per haplotype here, you're still achieving 96% completeness in your assembly. And it goes up a bit more with increasing coverage, but really where you see the rewards of more coverage is in the contiguity, or how fragmented your assembly is. So the tipping point, I would say, is between sort of 10x per haplotype and above. So 20x to 30x coverage is when you really start to pull things together into a relatively small number of contexts. And so lower coverage data sets will give you more fragmented assemblies. That tipping point of around 20 to 30x seems to hold true across a wide range of different data sets. But the other thing, I guess, to really emphasize is that the properties of your library are really crucially important. So if you can obtain high molecular weight, clean DNA, you will get better sequencing yields. And that higher N50 that you will recover really does carry through into your data. So these are two data sets, same species. One has an N50 of 40 kb on the left. The one on the right has an N50 of only 15 kb, which is still not a bad library. It's a bit of a lower coverage data set so that the sampling is sparser, but the important difference between the two is the total number of contigs that you recover. So at 20x coverage in the 40 kb dataset, you're looking at about 500 contigs, whereas you're up to around 2,500, 2,200 in at the same level of coverage, but with shorter reads. Returning to our assembly specifically, this is the data set that we put into the assembly. So genomic DNA was extracted from snap frozen young leaf tissue with an STS-based method. And we did do size selection using the PacBio SRE extra long reagent. And this is a selective precipitation that depletes fragments of under 40 kb. And the reason that we do this is really just to focus our sequencing effort on those long informative reads. We're using the LSK114 library prep, so the current ligation sequencing chemistry. And just 2 Promethean flow cells is the entirety of the nanopore data going into this assembly. We obtained about 180 gigs of sequence data, which is about 350x coverage, but we're not using actually a very large portion of that data set. What we are doing is filtering for quality and length, and then feeding specific subsets of that into the assembler. And the duplex, this highly accurate data, is really the backbone of our assembly. And I can just share with you that the duplex rate that we were obtaining on these regular flow cells was 27%. This is just an idea of a little bit more what it looks like. So this is the duplex portion on the left. You can see quality is really in that Q30 to Q35 space, which is just awesome compared to where this technology was just a few years ago. And we have about 48x coverage of this duplex data. There are nanopore ultra-long library prep kits. We didn't use that. We're just using our standard LSK114. But it's a bit sparse in the 100 kb plus range for this particular prep. So what we're using instead is what I term a quasi ultra-long set. So we've used a cutoff of reads bigger than 50 kb. And we've got about 75x coverage of that. And these are just simplex reads. They're slightly lower quality, but very useful for stitching things together within the assembly. This is, I guess, a flowchart of the assembly process as I see it. So there's read QC and filtering that happens upstream. We feed that data to the assembler. And then there's a kind of iterative process of curating that assembly, getting rid of things like retained duplication, polishing up the sequence quality, and evaluating as you go and tracking various metrics. And there's a lot of tools out there to do that at the moment. And I just really wanted to highlight a few of the ones that I've found particularly useful. So Chopper and Nanoplot are very good for exploring quality parameters of your long reads. There are a number of different assemblers out there that you can try, and the optimal assembler might differ for different genomes and different data sets, but Fly and Hi-Fi ASM I both find work very well. They're also very quick. Verco and NextInnovo are a little bit more complicated, require a lot more time and compute, and can be a bit harder to get running. Many of these tools have been developed for data sets that are predominantly vertebrate, and so it's quite interesting translating them into plant systems that are much more heterozygous before you even get to questions of polyploidy and so on. Purge dupes is a very useful pipeline for getting rid of retained duplication. That can be a real problem with these raw assemblies. YARS is good for scaffolding high-C data. And I have included Minimap2 because a lot of these processes involve mapping reads back to draft assemblies. And really just to highlight that in the last couple of weeks, Minimap2 has had release updates where they have implemented models that are optimized for these highly accurate long reads. Number of different tools that you can use for polishing, some of which will work when you've got a mix of data sets and some of which will just take ONT data only. And then a bunch of tools that are useful for tracking assembly stats. Mercury is one that's very widely used. It uses k-mers to evaluate things like accuracy and completeness, and is probably the main reason why we generated a short read Illumina whole genome data set as well, because the k-mer profiling of the nanopore reads is still not very good, because although they're very accurate, they're not quite as accurate at the level of short k-mers. And so we have that Illumina data set, and it allows us to really evaluate the quality of the assembly that we're bringing through. The assembly pipeline that we actually use, just to highlight the tools where I've presented options before. So the assembler that we've taken forward is HiFi ASM, and we've used it with the Raft extension, which I'll go on to explain in a couple of slides. I use NextPolish 2 for polishing, which uses the Illumina reads to identify repetitive regions, but actually polishes with the duplex reads. And we have done Hi-C scaffolding, but really is an evaluation tool. So things are actually coming out very complete from the assembler. And so we haven't needed to use it to pull things together, but we've used it really as a sanity check for what's coming out. There are increasingly assembly standards out there for which allow you to basically place your assembly on this metric of where things were. So BGP type assemblies is probably a good assembly 2-3 years ago, and a completely finished assembly up here. And so the more to the left of this table we arrive, the better the assembly that we have. And these take on what's traditionally been called the 3 Cs, so the contiguity, the correctness, and the completeness of an assembly. So, and there are different tools that you can use to evaluate your assembly against each of these. HiFi ASM is the assembler that we've used, and it's got a couple of really neat features. So it can take these ultra long reads. This is this double graph method in the paper at the bottom left. So which means that you build a backbone with your highly accurate duplex reads, but then you also integrate that graph with one that's slightly noisier, but should be able to get you through gaps and repetitive regions. And you can also feed Hi-C data into that assembler at the outset. We have Hi-C because we have a long-standing collaboration with people who are very good at that technique. In principle, you could use poor C data to achieve the same aim and have a purely nanopore level of chromosome conformation capture. The Hi-C at this point in the algorithm, it's not scaffolding anything together. What it is doing is helping to resolve the phase. And so essentially you recover haplotypes that should have less phase switching than if you hadn't provided this information. I said before that I used the Raft algorithm. This was published late last year. And it's designed to deal with the problem that occurs when you've got reads of variable length within your library. So it's particularly pronounced in nanopore when you can have a really long tail of read lengths, and you get contained reads in the middle that kind of lead to assembly gaps. And so what this process involves is essentially running the assembler to error correct your reads as you would normally do, and then taking it through a process where it then fragments those reads so that it deals it generates a smaller set of inserts, but ones that are still capable of mapping over repetitive regions. And I was finding I was getting quite good assembly quality without the Raft implementation, but usually kind of 2 or 3 contigs per chromosome, whereas the moment that I incorporated Raft, all but one of my chromosomes came out as telomere to telomere. So this is a kind of obligatory assembly statistics table. So I've got the 2 haplotypes. They're both around 500 megabases in length, the same number of contigs. So there are 19 chromosomes in grape. I have 18 chromosomes as single contigs, not scaffolds, but contigs, and 1 chromosome that comes out in 2, and the others are the organelles. And interestingly, the mitochondrial genome has been particularly troublesome, and that's still present in as 2 contigs in the current draft. We can look at how those haplotypes compare to the established grapevine reference, which is a Pinot Noir inbred line sequenced to very high quality in France. And broadly, we're really quite synthetic with them, but there are differences. So this here is the unmapped contigs, but broadly we have single contigs per chromosome, and the alignment's pretty good and maybe a little bit wobbly around the centromeres, which is probably where you would expect it to be. So this is just an example of the Hi-C contact map. So it actually hasn't been normalized, but it's a really pretty good recovery of the chromosomes. And in terms of completeness, we've got very good completeness. The level of duplication is the same as we see in the standard reference, so it's probably real. The genes that we're missing, there's 2 of them from the eudicot set, and those 2 genes are also missing again in the primary grapevine reference. It's highly heterozygous, as I mentioned, so while the individual completeness of the k-mers is only around 80%, when you put those 2 haplotypes together, you get to over 99% completeness. And this QV score of over 60 is pretty good going. So this is a polishing, I did a polishing step that did include Illumina data. Even before polishing, I was in the greater than Q55 for QV. So in terms of where we're tracking, it's looking pretty good. So we still have a few criteria to tick boxes on, but broadly we have a really nice diploid assembly that's very valuable for our project. And so just in the last few minutes, I'll give a little bit of a flavor of some of the things that we're looking at with that assembly. And transposable elements, I mentioned that that's what we're trying to mobilize within our soma clones. And so we really need to look quite hard at what the transposable element complement is within these genomes, and what the landscape looks like. And there's a whole bunch of different tools that you can use to do that. And I've been looking particularly at EDTA, which is quite widely used in plant genomics, but also more specifically about how important it is to provide any kind of curated reference set. And so without any kind of curated library, just going through the standard pipeline, these are the proportion of retro elements, DNA elements, and unclassified that we see, and just the total repetitive content that it reports. And I've tried also providing the same pipeline, but with a curated library and from a number of different sources. So one from the official Grapevine reference, one transposon library from the UC Davis, from Dario Cantu's group, and also one from the Plant TE database. And probably all of these do a little bit better with the DNA transposons, and that's maybe not surprising because I think it's understood that EDTA does less well with the DNA elements, but still a pretty comparable recovery, at least at this level of the different elements types. And particularly, there's not a massive enrichment of unclassified elements within without providing a curated library. The repetitive elements we can also use to look at the quality of the assembly using what's called the LTR assembly index, which essentially looks at how often you recover both ends of the LTR intact. And these are the numbers that are reported. Essentially, the higher, the better. Where it actually lands will depend on the properties of your genome, but the conventional reference is around 16. There is a PacBio Sauvignon Blanc genome that was published last year. That's sitting quite a bit lower, whereas our 2 haplotypes are sitting a little bit above the conventional reference, and that's encouraging for us to see. This repetitive portion of the genome, in my opinion, is the part of the genome that nanopore datasets do particularly well in. So we see this burst of elements here, which when I look at the PacBio genome that I mentioned previously, doesn't appear at all. And I have a bit of work to do to really drill down into what that is, but my working hypothesis is that these long reads in the nanopore datasets are doing very well at getting through those difficult parts of your assembly. And I'll just go on now to really talk about how we can use this assembly for these clones that we're generating. This is just a picture of our plant growth chamber. And I guess the way that I think is quite compelling for the real value of this assembly is that when we map our data sets from 5 different commercial clones that are grown to the conventional plant reference genome, overwhelmingly what we recover is on the right here, and it's about 55,000 structural variants. This is before any filtering, but it's a large number of structural variants, but they're shared between all of the Sauvignon Blanc plants. And so having those in our data sets when we look at scale at these genotypes is really a bit of a distraction. If we do the same when we're using our own reference, and this is a reference from about 6 months ago, and we've moved things on a little bit, but the point still stands, and that is that we then really focus our effort on variants that are interesting. And we start to see things like this plant here has the largest number of unique variants. And actually this clone has a very different import history. Whereas the sharing of these here actually fits again with what we know about their import history, and we would expect them to be more related to each other than other
Yang
plants.
bisa apa aja?
We can also see the advantages of long reads when we look at mapping. So if you compare the same region of a genome between nanopore and Illumina, Illumina, you see a coverage dip here because this is very, very different for this stretch of 1.5 kb. And you just can't map those Illumina short reads, whereas with the long reads, you've got flanking information. And in fact, you can recover that quite well. Similarly, here we have an insertion of 850 base pairs. In the Illumina, there's a little bit of a hint that you've got some reads that don't have the right expected insert size or that are missing a mate. Whereas in the nanopore data, you know exactly what it is that's different in this heterozygous variant. And I think I'll wrap up there and just really say that this is a very exciting time to be using nanopore sequencing to assemble plant genomes. We're obtaining near gapless, telomere to telomere, phase diploid assemblies of highly heterozygous plant genomes, and we're doing that in a way that is very cost effective. So only 2 flow cells is enough to get us to that point. And with that, I will just acknowledge the people that I work with, our team at the Brigato, also collaborators at Plant and Food who are involved in the plant production work, and a lot of the informatics support that we get from Nessie and from Genomics Aotearoa. And I'd be very happy to take any questions. Thank you.
Yeah, thank you, Dr. Weebly, for the insightful presentations. I think it is really interesting to see how you assemble the data of the grapevine. I think it's quite a huge data set, right? Because you have this coverage of 300s. Probably you need to do this in like HPC or something, because I could imagine how big the data is. And then for the participant, I think I would like for you to ask, because we will have the direct Q&A for Dr. Weebly. So if you have any question, you could raise your hand or maybe put it in the chat box. Okay, I think Ibu Lina have a question from PRkin. Yeah, silakan Bu Lina. Yeah.
Thank you very much for Annabelle. It's a very good presentation. So what I would like to ask is about the reference. So you are using the Sauvignon Blanc plant as a reference. So I was doing the transcriptome analysis using the reference from the before you adding the PN40024 version 4 from the France group. No, it has been published yet. So I really think, do you think that because your presentation mentioned that your assembly is better than the previous one, so do you think that your assembly can be used for another cultivar such as like Shiraz or Chardonnay, something like that? This is the thing that I need to reconsider whether or not I need to analyze again the data set or whether I stick to the past reference. Thank you very much.
Yeah, can I ask where you were doing that work?
Where?
Yes.
Yes, this is data from the Uni Adelaide data set. So it's actually from other PhD student. I was graduating, I graduated from Uni Adelaide in 2022, but I'm not working on this grapevine. I'm actually working on other plant assembly as well, Plantago. Yeah. Thank you.
Yeah, I ask because actually we have collaborators at Plant and Food in New Zealand who have just asked us exactly that. So they have a historical Sauvignon Blanc RNA-seq data set, which they just couldn't make sense of with the version of the reference, again, before PN 4024, version 4. So a very similar situation. So in a few months, I would know exactly how much it would make a difference.
Okay.
I think it would probably still be worth revisiting because the assemblies have come on so much. We can map like 99% of our reads will still map to the non-Sauvignon Blanc reference. So it's not a terrible proxy, but I think that there might be regions where things are more complicated, where having a better reference would help. The only other thing I would add is that it's kind of interesting to me. So the official reference, when they annotated it not with transcripts from that reference background, but from a whole variety of different cultivars and also different treatment conditions and so on. And so I think there should be quite a lot of translatability between, but like you say, I've also heard that it's not that straightforward in practice.
Thank you very much.
Thank you, Marina. And then I would like to ask again for the participant if you have any other questions. Bapak Ibu, dipersilakan. So if there's not any, I actually have a few questions. The first one is that about the extraction. We know that for the ONT, it's really difficult. It's really important to have a good library, right? Because, for example, you have like small fragment, it will hinder the sequencing process. And then you mentioned before that you are using the the SDS-based extraction. I mean, we tried several times using different DNA kit extraction, but it's not really working for us. And also sometimes people here tend to use this CTAB extraction. And how is it about with your experience? Is it like SDS-based method is the better ones, or do you have any other preference for the extraction process of the plant?
Yeah, we've tried a few different methods, and I would also say that it can be really species or even cultivar specific. So certain plants have different secondary compounds and different methods to perform differently on those species. We use the Mascherinagel magnetic beads protocol when we're doing extractions at scale, and that also works pretty well for us. We have tried CTAB-based approaches. It's just that kind of for historical reasons, it's the SDS one that we kind of really optimized in the lab. There are a whole range of kits out there. When I was doing, so the bird work was kind of interesting in that respect, because birds they form a lot of G quadruplexes, so they're very blocky samples. They're quite hard to get yield out of. And we found there was a real compromise in extraction methods. So I think the NEB Monarch long high molecular weight kit with the glass sort of balls gave us really nice molecular weight profiles, but it wasn't cleaning up the sample well enough. And so although we could get long reads, we couldn't get very many of them, whereas the PacBio, the old circulomics kit was much less blocky, so was really our kind of best choice. But we also found that standard like the QIAGEN is also very good with the genomic tips, and also kits that are not necessarily marketed as high molecular weight often will still give you a good proportion of long fragments if you're careful with your processing. So I think it's really worth the time to optimize that extraction protocol.
Yeah, I agree. Yeah, I agree also that different kits offer different positive impact, because some kits it is really good for the high molecular weight, for example, but not really good. The purity is not really good, like you said. But the other kits offer different things, you know. Yeah, I think we need to optimize the kit also, like you said. And also, what is the good coverage if you are thinking about plant assembly? Because you mentioned that like the first data you see that from the coverage of 20 to 80, you have less context, and it is good because you want it to be like one big genome, right? And what do you consider the good coverage for a plant?
So I think if I were targeting a coverage, I would be looking for 40 to 50x coverage. I think that's a really that lands you very comfortably in something that you'd expect to be quite contiguous. But it gets very expensive when you have very large genomes, and so there's a bit of balancing to do. But that would be my starting point. And if I could get that in duplex data, then that would be great. If you only have simplex data, you're still going to get really very nice assemblies.
Okay. Yeah. Okay, I give it back to the participant. If you have any question for Dr. Weebly, is there any question? Oh, I think it is very clear, the presentation, but maybe I would like to ask for Dr. Weebly for the recommendation or like a good practices when you are do this plan whole genome sequencing. What things you need to consider and what things you need to be careful of if you have comments on it, like general comments on it.
Generally, I think that it's a really exciting space and there are lots of developments. So just really being aware of, so nanopore very often improvements in Dorado and can make a really big difference to your data set. I think really just keep sanity checking what you're doing, essentially make sure you've got tools for evaluating and I guess the other thing that I would say is that also bear in mind what you need your genome for, because you could spend a very long time finessing a genome and it wouldn't materially be any different in terms of what you actually want to use it for. And so there's a little bit of like, you can make some very big improvements early on and then it sort of slows down. And I guess in terms of projects, that's something to be mindful of.
Okay then. I guess thank you, Dr. Weebly, for the very insightful presentation and also fruitful discussion. And we are really happy that we are meeting with you today for this webinar. And then hopefully we will meet later, probably for discussion if we have any question for you. So thank you again. And then next on our agenda, we have our second speaker, Dr. Chen Liao. She is also from Bragato Research Institute. So Dr. Chen Liao is a scientist of grapevine epigenomics in the grapevine improvement team, so also in the same team as Dr. Weebly. So Dr. Chen Liao primarily works on the Twin Vines project to study epigenetics of grapevine in different climates in response to stress. I think it is already conveyed also from Dr. Weebly. And Dr. Chen Liao will present on the ONT methylation sequencing for grapevine epigenetics profiling. I think I already saw that Dr. Chen Liao is already here. So could you hear me, Dr. Chen Liao?
Yes, Dr. Sarah, I can hear you very well.
Okay. Yes, thank you. And now the floor is yours.
Thank you. I will share my screen. Can you see the slide?
Yes.
Thank you. Selamat pagi semuanya. Saya originally from Jakarta, tapi sejak 7 tahun lalu I'm in New Zealand PhD, lalu lanjut kerja di sini. So first I thank ONT and teams at BRIN for the opportunity to present our work here. As Dr. Sarah has mentioned, my work is on grapevine epigenetics. So today I will talk about how we use ONT to do DNA methylation sequencing for this purpose. So this is the outline of my talk. First, I think I would share briefly about plant epigenetics. So we probably all know that epigenetic regulation is an important part, both in plant development as well as in their response to any environmental stress. So in plant development, one of the most well-known example is probably how DNA methylation changed during fruit ripening, as shown firstly in tomato, where during fruit ripening, the pericarp methylation undergoing global demethylation, resulting in hypomethylation. And this is also has been shown in other fruit, like strawberry, that show the same pattern as tomato with a global hypomethylation during ripening. And in another fruit, such as sweet orange, there's an opposite pattern with a global hypermethylation during ripening. And disruption of this methylation process will delay the fruit ripening that show a causal effect of the role of methylation for this process. And in terms of response to environmental stress, because plant can't move, so when exposed to various stress such as extreme heat or droughts, or differs pathogen and pests, they have developed a system to fight these threats, including the epigenetic regulation, such as DNA methylation, histone modification, and also the involvement of RNA silencing. So when we talk about DNA methylation in plant, it is quite unique compared to what normally occurs in invertebrates, such as in human, where DNA methylation in the DNA sequence normally only occur at the CG context, as shown in the left part here, where in human, methylation only occur at C preceding a G context in the DNA. But in plant, methylation at cytosine can happen in any context, and they are being regulated by different pathways, and they are involved in different biological processes. So they are normally grouped into three different contexts. So CG, such as in human, and CHG or CHH methylation, where H here is any base other than G. And although less well studied in plant, but most well known in species such as bacteria, 6-methylated adenine has become more and more studied. And in plant, it has been shown to be enriched in genic region and associated with higher gene expression, in contrast with methylated cytosine, which is normally associated with repress of gene expression. So why we use nanopore sequencing for this purpose? So traditionally, this resolution DNA methylation is maybe most commonly done using bisulfite sequencing, where we need to treat the DNA with a chemical called bisulfite. So methyl bisulfite will convert any unmethylated cytosine into uracil, and upon PCR or amplification, it will translate into a thymine. While methylated cytosine will not be affected by this treatment, so after PCR, it will remain as a cytosine. In this way, we can differentiate whether a particular cytosine is being methylated or not. This process is quite established, but nanopore sequencing detects DNA methylation in a very different way. So Annabelle has explained the basic of nanopore sequencing, and this applied also to modified base, because each base, when they go through the pores, they disrupt the signal in a certain pattern. And because modified base have different physical properties as well, although they are the same base, they also disrupt the signal in slightly different ways. And with the base calling algorithm, it can be translated whether that base in a particular position is being methylated or not. And this is translated as a probability score of the occurrence of the methylation. So it offers several advantages over the traditional methods. First, we can detect this methylation simultaneously with any canonical base. So when you do nanopore sequencing, you can obtain this DNA methylation sequence or signal at the same time, as long as you activate that mod-based model on the amino interface. So you don't need any additional sample pretreatment, you don't need any additional cost to obtain this additional information. And because we do not need any DNA treatment, such as bisulfite, which is known to degrade DNA, we can preserve the longer DNA fragments, which can result in better mapping and, of course, higher accuracy. And because we can use ONT data for phasing or to obtain a separate haplotypes, we can also obtain haplotype-specific methylation signal for each chromosome. And unlike bisulfite sequencing, which only detect 5-methylated cytosine, nanopore sequencing at this current chemistry and technology can also detect hydromethylated cytosine as well as methylated adenine at the same time. And when we combine this with sample barcoding, we can, to multiplex multiple samples in one run, we can result in quite a cost-effective method to detect methylation. So this is a workflow of how we get the methylation signal out of the ONT data. So normally we result in port 5 out of our sequencer, port 5 raw data, or FAST5. We can convert that into port 5. And normally we rebase call this using Dorado, just because usually the amino interface do not have the latest base calling or modified base calling models. So if you rebase call with offline Dorado, you can use the latest, which is usually the most accurate model available at any time point. So you can use a model to call mod base of 5MC and 5FMC at the CPG context only. This is probably predominantly used for like human genome, but for plants, we can use 5MC and 5HMC in all context cytosine, and also the 6MA modified base model. This all will be recorded in our resulting BRIN file out of Dorado with the scores called MM and ML scores in the BRIN files. And when we take this BRIN file, convert it back into FASTQ, and remap them using tools such as Minimap to our reference, we can then extract the methylation signal into a tabular form using a tool called ModKit, and result in this tabular file called a bed metal file. So it looks something like this. So we can get at any particular chromosome position. We can see what modified base is being recorded. Here is M, which represent methylated cytosine, and 6MA is normally written as a small a letter. And we can see how many coverage of reads in that particular position and how many % of them are being methylated. So when we first started generating data of methylation sequencing from ONT last year, when the first model for plants just emerged for the R10 flow cells, we first want to see how well the data correlated with the more traditional bisulfite sequencing. So as shown in the graph here, that both data set correlated very well with the R value of more than 0.9. And I think there are opinions out there that nanopore sequencing methylation is more accurate, but we can really prove that which is more accurate, but they correlated very well. And the other things that we did is also to determine how many coverage we need to still generate accurate DNA methylation detection. So I used the same dataset as Annabelle used for her assembly, which is generated from Sauvignon Blanc grapevine sample, sequence using LSK114 on R10 flow cell. And I took one sample, which is 120x coverage. Then I subset them into lower coverage of 60, 40, and 20x. And when correlated this dataset with the whole genome bisulfite sequencing, we can see that the correlation remained quite stable from the high to low coverage. And when we plot this methylation data across, for example, here across chromosome 1, we can see that the methylation data or methylation level detected is quite stable across all the different coverage. So we can conclude that with around 20x coverage, we can obtain an accurate DNA methylation data of this data set. So once established the technical performance of DNA methylation detection with ONT, we use this in our grapevine project. The first project I did with this ONT DNA methylation is to map the T-RAW effect on grapevine. So T-RAW is a concept maybe mostly known in grapevine, where grapes that grown in different locations, affected by different soil type, different climates, and different plants and people management of the plant will result in a unique characteristic of the fruit and the resulting wine. But this concept is actually extended to other crops such as coffee or cocoa or tea, where specific location crops has a unique characteristic. So to do that, as Annabelle has mentioned, the Sauvignon Blanc in New Zealand are genetically homogeneous. They're all the same cloned and being propagated clonally, and we basically grow the same genetic material throughout New Zealand. But however, the Sauvignon Blanc from the middle region here in the north part of South Island, called Marlborough, they are well known for their very distinctive wine characteristics. So we want to see whether, although they have the same genetic material, they result in different product characteristics, whether DNA methylation or epigenetic contribute to these differences. Most of this plant has been growing for about 20 years when we sampled them. So we collected a total of 48 samples from 16 different vineyards from these 3 different regions, and we sequenced them each to around 20 to 30x coverage across 8 flow cells. So this is just an example of a PCA plot on CG methylation we see we can cluster them very nicely. So the one in green here is the grapes grown in the north part of New Zealand here, and the red here on the right is the one grown in the southeast part of New Zealand at Otago. And the one from Marlborough is actually can be subclustered into two different clusters, the one in purple and the one in blue here. So we can see that although they are the same plant, they are epigenetically quite different. The other things that we have working on is develop this or validate most of this method called SkimSeq, which is a very low coverage sequencing, normally below 1x coverage for methylation, for global methylation profiling. This is based on a paper published last year by Christopher Fogg. He used this method, which he designated genome skimming, combined with nanopore sequencing to precisely determine global and transposon DNA methylation in vertebrates. So an example of the graph that he presented paper is a chimpanzee genome here, which he sequenced to 10x coverage, that he can obtain the CG methylation level at around 82% here. But when he downsample this data set down to 0.1x here in the orange, the global methylation level remained very stable. So to detect global methylation level, you actually don't need high coverage, but you can only need like 1% of the genome is enough to still obtain the same global methylation level as the high coverage data. So to see whether we can apply this method to our grapevine data, we apply the same method. So I subsample a high coverage Stephanom blanc sample down to 0.01x with 10x replicate bootstrap, same as the paper by Fogg. So we see here that it's quite different to the result in vertebrates. We actually need a slightly higher coverage. So at 0.01x as the reported in vertebrates, our variation is already very high. So we cannot achieve the same accurate global methylation anymore, but we can achieve the same accurate at 0.1x or 10% of the genome. And given grapevine genome is not that large, this is still quite a cost-effective method to look at global methylation level. And to see whether this pattern is consistent across all plant genomes, across several plant genomes, and not just characteristic of grapevine, which is highly heterozygous. We perform this method on two other plant species, one model Arabidopsis and another species Actinia melananda, which is a species of kiwi fruit. So we see here that actually the same, and we also compare it with publicly available human data from ONT, sequenced under the same chemistry and flow cell. So we see here our human data set resulted in a similar pattern to that paper. So we can obtain an accurate assessment of global methylation level at 0.01x. But for the three plant species, it is only accurate down to 0.1x. So it is quite consistent. And we can also use this method to look at specific genome features methylation, not just global. So as shown here at the 10x coverage data, we can see the pattern of, for example, CG methylation, which is high in transposon element region and lower in genic region. And this pattern is still highly conserved when we only use 0.1x sample. Although we can see the variation is slightly higher, but we can still have the same profile. We assess what are the technical contributors to this higher error rate to be able to see if we can pin down why the plant genome is more variable, but we haven't succeeded in that yet. But we did show that several technical factors contribute to the error rate, which is higher, such as the first one is read length. As Annabelle mentioned, our data set is relatively high long reads because we already removed all the small reads. But if we subset the samples or trim down the samples to shorter reads, here to 5 and 10 kb, we can show here in the dark gray that actually the error rate becomes lower compared to the reads, which is very high length here in 50 kb. We also try to compare, because my data set is being mapped to the PN version 4 reference, because I started way earlier. But we show that if we use our Cephinum Blanc assembly that Annabelle generated, we can also see a better accuracy if we use a better reference genome for our sample. And this method can be used for multiple purpose. It has a potential to screen for mutation changes upon, for example, stresses or across different development stage in plant. One thing that we have used here is shown in this graph here, where we try to see whether the DNA methylation is stable across tissues. This is because when we sample, like for example, grapevine, we sometimes sample different size leaf, depends on the availability of leaf in the field, and we want to know whether we can still get a consistent DNA methylation level, even though we have slightly variable in the leaf that we collected. We show here that for CG and CHG methylation, the methylation level remain very constant across 3 different leaf size, and also in the tissue, which is tendril that we collected. But for CHH, the tendril methylation level is actually significantly higher compared to the leaf tissues. So that summarized my talk, and I would like to acknowledge the people in my team who has all contributed to the work here, and also for the funding bodies, including the New Zealand MB, and supported by NESI for facilitating our bioinformatic analysis, and all the growers and industry partners who contributed funding and also the sample for this work. Thank you.
Thank you, Dr. Chen Liao. So I think it is very interesting talk about the methylation sequencing especially. And I would like to ask for the participant to ask any question if you have some question. Okay, so I have question. Oh, I'm sorry. Okay, Balina is also have a question. I think you could start, Balina.
Thank you very much, Mbak Zahra. Thank you very much, Bu Yusmiati. Can I call you Yusmiati?
Of course.
So the last slide mentioning that the size leaf, so there are correlation probably between the methylation profile with the size of the leaf. What's actually the meaning of this data? If we can connect to the, what is it, theory or a product of the grapevine itself? I mean, the taste or the anything at the final product. Thank you very much.
Thank you for the question. I'm not sure that I understand the question, but I will try to answer. Betul dikoreksi kalau salah. So what we show in the last slide here is actually the methylation in the leaf. Although they are different sizes, are quite stable. Like although here in CHH is slightly higher, but they are not significantly different. The tendrils show highly methylation compared to leaf in CHH. Tendril is like our control, like to see whether we can detect changes in the methylation level. But across all the leaf size, they're actually quite stable, which is quite reassuring because sometimes you collect like different sizes, depends on what's available in the field.
Okay.
Yeah. So we haven't tried to track them to the resulting end product yet, but for the one that I show here, for the Terra study, like we see that different regions can have different methylation profile, and we actually associated with some of the grapes quality, like the grape juice analysis. Some of them show a significant association with the DNA profile. But because our sample size are quite limited, there are only 48 samples, it's quite hard to make a very strong, powerful association with all the phenotypes because they are quite different other than the region. They have different vineyard management, they have different rootstock they are planted on, etc., etc. But there are some potential association with the grapes, the resulting grapes from each region.
Okay. So yeah, I
To
just
answer the
wanna,
question.
yeah, I think so. I think this answered the question, because what I really want to know why you are doing DNA methylation, because you mentioned in the previous slide that it's related to the what is steroid? I don't know what is the taste, the lane. I mean
Yeah.
the management, something like that.
Yeah.
So
Yeah.
I'm guessing that you are doing this because you wanna know if there is, at the end we can predict from the DNA methylation to actually predict the final product, something like that.
Yeah, maybe if we have lots of samples, they are more controlled, we will be able to do that. This is just a very early pilot profiling study. Yeah, and the aim of this profiling is to see, this is part of the project we call TuneFind, is present in the enable slide, that we want to see whether we can modify these traits by using RNA, if we can identify the target or where the changes happen associated with any trait, to see if we can use RNA to silence any specific part of the region.
Okay. Thank you very much.
Thank you, Marina. So I have also another question regarding this one. If you could put a scale on it, how big is the difference in the DNA methylation between different area? Like, could you put the difference in the number of methylation, for example, between these different places?
Yeah. Yeah. So other than the PCR port here, we have detected several differentially methylated regions, like the part of the genome that are actually difference between the two geographical regions here. I can't remember on the top of my head, but I think we detect about they are mostly at the CG context. They are mostly different. CHH and 6MA are not quite different. CG is the highest level, maybe about 50 methylated region. And the region that we define here is 1 kilobase size tiling window. So we compare it windows to windows with about one kilobases. I'm not sure how that scale up compared to the genome, but yeah, we're still trying to analyze what that biologically, what that means biologically.
Okay. And then another question is, you mentioned that before the shorter reads is better for the investigation of the global methylation, right? Because when you take a look into the largest fragment, you see that the error is higher than the shorter fragments. Does it mean that if you are looking into this global methylation, is it better to use like a short read sequencing? Because later, I mean, if you would like to have shorter, then probably long read is not really preferable for it.
Yeah, it's a relative term like short and long here. I think we don't want very, very long reads. Like we don't want like 50 KB reads, just because we don't sample this, right? We have like only 0.1x. So some region will have some reads covering it and some might not have. And we have long reads, then the randomness, like one read like 50 KB can have different methylation level, right? Like one part can have like high methylation, one part have low methylation, and they kind of like collapse together. But if you have shorter reads, they are quite independent. That's why they are more accurate when you randomly down sample them. Yeah, that's why it's quite preferable to have like shorter compared to very long.
Because you randomly choose the fragments, right? And then it will be more representative if you have shorter fragments for that.
Yes.
Okay. Okay, all is clear. And then I will give it back to the participant if you have any question for Ibu Yusmiati. Okay, maybe also I have another question about the base calling. I mean, usually when we are using ONT, we are using GAPI, right, for the base calling. I think this is the one that are bought by GENOME software.
I think in GENOME, GAPI has been replaced by Dorado as well since last year. And GAPI is basically being stopped developed. They develop all their new like base calling models and modified base model on Dorado. So I think GAPI is still available if you want to use them offline, but they are not updated with the latest model anymore. So if you want to
Okay.
get the latest, the most accurate model available, it's better to use Dorado.
Okay, so then, yeah, because we are usually, I think we still use GAPI in here. I'm not really sure about the Dorado, but we will take a look into it. So
Yeah,
I guess
Felicia
thank
and Fania
you.
can answer it. I think we updated our Mino like last year. It's all using
Okay.
Dorado. But
Okay,
Felicia
okay.
and Fania can answer too once installed.
Okay.
Yeah.
Then, okay, so Ibu Fani already commented that let us know already use Dorado, and probably we need to update, I think. Okay, so all is clear. Thank you, Ibu Yusmiati. If you have any last comments on the participant that would like to work also on this global methylation investigation, do you have any suggestion or like some kind of comments for the researcher here?
Yeah, this is just an additional data you can get at basically no cost at all when you do your nanopore sequencing. So why not use it if you can think of a way to have your data analysis or to your project, because it's free if you do any nanopore sequencing.
Yeah, because I think it is very interesting also to make a connection between this global methylation to the physiology of the plants. Also, like you mentioned before, for the taste of the grape, for example, I think it would be very, very interesting to take a look into that. Yeah. So thank you, Ibu Yusmiati, for the insightful presentation and also for the discussion. It is really, I have a lot of information regarding this global methylation sequencing, I guess. And then hopefully we will meet later if we have any question to you regarding this, because I think some researchers are also interested to take a look into this global methylation sequencing. So thank you, Ibu Yusmiati. Selanjutnya, Bapak Ibu, kita kedatangan Ibu Dr. Ratih Darmayanti. Ibu Dr. Ratih Darmayanti beliau adalah Direktur Pengelolaan Koleksi Ilmiah BRIN dan akan membawakan presentasi terkait platform kolaborasi riset akuisisi data keanekaragaman hayati dan non-hayati di Indonesia. Jadi di sini nanti juga akan dipaparkan terkait dukungan layanan sequencing yang diberikan di lab sequencing BRIN. Jadi banyak informasi terkait lab sequencing jika Bapak Ibu bekerja di sekitar area sequencing. Untuk itu, saya undang Ibu Ratih untuk menyampaikan paparannya. Saya rasa Ibu Ratih sudah di sini ya, Bu.
Ya, makasih Mbak Sarah.
Ya, silakan Bu.
Yeah, thank you very much, Sarah. And the first speaker is still here, Dr. Annabelle? Already
Yes, I'm here.
Oh yeah, sorry, Annabelle. According to the committee's request, they asked me to talk in Indonesia, but want to just give you some introduction that what I want to talk is about the program in Indonesia that we want to develop one data for biodiversity. It is including the whole genome sequencing of plants, animals, also humans in Indonesia, and what you already presented here very support the program, and we hope that you can also join the project here. You can have a counterpart with maybe Sarah or Indonesian researchers, and then we want to your help or your support in these programs. And thank you very much for Dr. Yusmiati for the presentations. Like Zahra said, that what I want to present here, it will be supported by all of the not only Indonesian researcher but also foreign researcher, international researcher. And I'm sorry to talk Indonesia because all the audience here is Indonesians researcher, and Dr. Rifki asked me to prepare presentation in Indonesian and I will talk Indonesians. But if you have questions, we'll be very happy to answer in English. Yeah, can I start, Zara? Okay. Yeah. Ya, thank you very much for the committee. Makasih panitia sudah mengundang. Jadi ini adalah mungkin paparan pertama dari program platform kolaborasi untuk BRIN investasi strategis yang rencana akan dilaksanakan selama 3 tahun ke depan, yaitu akuisisi data sumber daya hayati dan non-hayati Indonesia. Saat ini yang kami identifikasi, termasuk juga arahan dari Kepala BRIN, bahwa untuk manajer program adalah di bawah Direktorat Pengelolaan Koleksi Ilmiah, karena sumber pertama kita untuk melakukan akuisisi data adalah berasal dari koleksi ilmiah yang saat ini ada di bawah BRIN, kemudian di situ juga akan terlibat PIC untuk infrastruktur dari Direktorat Pengelolaan Laboratorium, kemudian PIC untuk komputasi dari Kepala Pusat Riset Komputasi Dr. Rifki. Untuk PIC data ini ada Direktur RMPI, repositori dan juga Kapus Datin untuk database Sekahati. Kemudian dari tim BRIN saat ini memang masih kami koordinasi dengan teman-teman dari OR Hayati dan Lingkungan, OR Arkeologi, Bahasa dan Sastra, OR Kesehatan, OR Kebumian dan Maritim, serta OR Pertanian dan Pangan. Ini yang kami harapkan semuanya dapat terlibat gitu ya untuk mendukung program akuisisi data Satu Data Indonesia untuk sumber daya hayati dan non-hayati. Jadi Bapak Ibu di sini sudah ada di website pendanaan risnovbrin.go.id mengenai call for proposal untuk riset inovasi Indonesia Maju atau RIM investasi strategis. Di sana ada 10 tema. Bapak mungkin sudah tahu ya, dari keragaman hayati terestrial, kemudian marine, ekspedisi, hingga yang nomor 6 ini adalah akuisisi data sumber daya hayati dan non-hayati Indonesia. Kenapa meliputi 2 tadi? Karena di koleksi kita, nanti akan saya jelaskan kemudian, itu ada koleksi hayati maupun non-hayati seperti batuan dan arkeologi. Jadi skema pendanaan RIM investasi strategis itu adalah pendanaan RIM untuk institusi. Jadi untuk melaksanakan kegiatan riset inovasi dengan tema yang mengacu pada rencana strategis BRIN. Tujuannya adalah untuk pembangunan pengembangan teknologi, kemudian sumber daya manusia unggul dan penguasaan iptek, serta sebagai modal utama salah satu pilar dalam pembangunan nasional. Nah, kata kunci dari RIM investasi strategisnya adalah kolaborasi, baik sumber daya periset maupun mahasiswa yang ada di Indonesia, di BRIN, di perguruan tinggi, di kementerian, kemudian juga di sana ada mobilitas periset, kan ada visiting researcher, postdoctoral assistant dari teman-teman perguruan tinggi dan juga nasional maupun internasional. Di sini dengan adanya pendanaan skema untuk mengkolaborasi ini, itu BRIN akan akan memiliki joint funding gitu ya dengan mitra luar negeri untuk melakukan riset bersama-sama. Dan BRIN juga di sini akan bertugas sebagai penyedia infrastruktur lab, kemudian untuk fasilitas mobilitas periset di bawah SDMI dan juga melalui pendanaan dengan Direktorat Fasilitasi. Dan dengan kegiatan ini kita juga akan manfaatkan 8 skema mobilitas periset, termasuk tadi sudah disebutkan seperti RA, DBR, dan seterusnya. Serta ini akan dibuka sebenarnya sepanjang tahun begitu ya untuk kegiatan tim investasi strategis. Tapi nanti kita akan memiliki timeline tersendiri di mana kami berharap teman-teman juga dapat terlibat dari awal. Dan ini kolaborasinya juga bersama mitra luar negeri melalui kolaborasi research, kemudian degree by research untuk S3. Nah, untuk RIM investasi strategis ini ada 3 kampus. Ini di mana mahasiswa dari luar negeri juga dapat mendaftar. Itu ada IPB, ITB, dan UI yang akan menjalankan sekolah bersama-sama di bawah supervisi dari periset BRIN maupun dari internasional. Kemudian sini juga memungkinkan ada pertukaran periset yang akan dibiayai menggunakan skema dari teman-teman di SDMI. Bapak Ibu, ini paparan saya ambil dari OR Hayati dan Lingkungan, bahwa ini kita bicara mengenai kehati saja Indonesia adalah negara mega biodiversity begitu ya, di mana kita dapat memiliki 34.000 jenis tumbuhan, kemudian 270 spesies amfibi dan seterusnya, di mana kita akan melakukan discovery terus-menerus, ekspedisi terus-menerus, kemudian di situ akan ada mekanisme penyimpanan di data ruangan koleksi ilmiah, di mana ini meliputi 3 hal besar untuk biodiversity, untuk kehati ada plants, animals, dan microbes, di mana nanti di dalamnya kita akan support dengan program-program seperti genomic data, protein, dan seterusnya yang tujuannya adalah untuk manfaatan yang berkelanjutan. Nah, Bapak Ibu, Ini amanah dari Undang-Undang Nomor 11 2016 mengenai Sistem Nasional Iptek dan juga Impres Nomor 1 tahun 2003 mengenai pengutamakan pelestarian kehati dalam pembangunan berkelanjutan. BRIN memiliki 5 tugas utama, yaitu melakukan inventarisasi, identifikasi, melakukan koleksi ilmiah, kemudian litbang di bidang data dan informasi kehati, serta eksplorasi bioprospeksi, dan juga bersama-sama dengan kementerian dan lembaga terkait melakukan koordinasi dalam rangka preparasi kebijakan berkaitan dengan pelestarian dan pemanfaatan kehati. Kita memberikan bantuan teknis dalam bentuk pelatihan dan konsultasi yang berkaitan dengan penelitian dan pengembangan di bidang pengelolaan dan pemanfaatan kekayaan hayati, kemudian melakukan pengelolaan kehati sebagai aset nasional dalam bentuk depositori dan repositori. Nah, di poin 4 ini, kita akan memanfaatkan aset tersebut ya, yang sudah tersimpan di BRIN untuk kita luaskan datanya sehingga dapat digunakan untuk pemanfaatan lebih lanjut, yang data-data tadi akan tersimpan jadi satu di repositori spesimen repositori di RIN tepatnya Pak Zainal. Kemudian ya teman-teman yang sudah lakukan adalah mengembangkan riset teknologi dan inovasi dalam pelestarian kehati. Nah, saya akan menyinggung sebentar nanti sekilas mengenai peraturan BRIN nomor 12 2023 mengenai wajib serah wajib simpan bagi data primer dan keluaran hasil riset, di mana nanti semua orang dapat menggunakan dengan mekanisme yang akan kita kembangkan. Nah, Bapak Ibu, dari amanah yang diberikan kepada BRIN, kita memiliki koleksi nasional. Saat ini ada 8 jenis koleksi dengan total 12 koleksi yang sudah ada di BRIN yang sedang diintegrasikan maupun yang sedang akan diintegrasikan. Jadi kita memiliki koleksi botani atau herbarium, kemudian silarium, koleksi kayu, zoologi, koleksi bank biji, koleksi mikroorganisme, koleksi hidup di kebun raya, dan ada koleksi non-hayati yaitu geodiversitas dan arkeologi. Nah, ini akan menjadi modal kita untuk melakukan akuisisi data. Tidak hanya data-data yang ada di BRIN, tapi juga data kehati yang tersebar di seluruh perguruan tinggi, di seluruh lembaga-lembaga riset, baik swasta, begitu ya, yang ada di Indonesia. Yang kita harapkan kita memiliki satu data untuk Indonesia. Jadi baik data hayati maupun data non-hayati. Di sini kami memang, ini adalah proyek yang sangat besar, gitu ya, selama 3 tahun ke depan kita akan coba lakukan. Keterlibatan dari teman-teman periset di sini sangat kami butuhkan. Dan menyusul kemudian akan ada koleksi untuk riset koleksi hidup begitu ya. Ini yang diusulkan oleh teman-teman dari OR Pertanian dan Pangan. Kemudian koleksi di kebun raya begitu yang saat ini tersebar di 5 kebun raya. Adalah koleksi-koleksi hidup yang akan kita gunakan sebagai sumber akuisisi data dan juga perluasan data kehati. Bapak Ibu, tujuan ini masih berkembang. Sebenarnya ini adalah paparan pertama seperti saya sampaikan untuk platform kolaborasi ini. Kita akan masih terus berproses untuk melakukan perbaikan. Jadi masukan Bapak Ibu di sini sangat kami harapkan. Tujuan kita adalah menyediakan data sumber daya hayati dan non-hayati Indonesia yang komprehensif untuk kebutuhan taksonomi, forensik, dan muatan berkelanjutan. Jadi ini kami berharap sudah dapat mencakup semua koleksi yang ada. Jadi tujuan utama adalah memberikan satu data sumber daya hayati dan non-hayati Indonesia. Seperti yang sampaikan tadi strukturnya seperti ini, dan kita bisa melibatkan teman-teman dari kementerian, KLHK, KKP, Kementan, Kemenkes, dan semua juga yang terkait yang belum saya sebutkan, perguruan tinggi dan lembaga riset swasta maupun internasional. Ini teman-teman silakan merangkul semua mitra yang teman-teman saat ini sudah bekerja bersama-sama. Dan ini sekilas mungkin yang belum mengetahui yang dari luar BRIN. Jadi koleksi-koleksi tadi dapat kita manfaatkan. Pertama untuk memberikan data dasar, data-data yang dasar artinya kita memiliki jenis tumbuhan apa, kemudian kita memiliki tumbuhan baik dari berbagai lokasi di Indonesia, dari mana dikumpulkan, dari mana siapa yang mengumpulkan, di habitat seperti apa dan seterusnya. Itu data itu tersimpan di database Makoyana di kebun raya Indonesia. Jadi tidak hanya BRIN tapi juga kebun raya yang ada di daerah. Dan Bapak Ibu bisa menggunakan segala resource yang ada di kebun raya untuk melengkapi data yang ada. Jadi di sini ada 5 kebun raya dengan spesifikasi masing-masing dari dataran rendah basah, dataran tinggi basah di Bogor dan Cibodas, kemudian dataran tinggi kering dan dataran rendah kering di Purwodadi dan Eka Karya Bali, kemudian untuk di kebun raya Ciwideng ini berdasarkan ecoregion. Dan ini adalah jumlah koleksi yang ada di masing-masing kebun raya. Bapak Ibu bisa lihat ada sekitar berapa lebih dari 50.000 koleksi yang itu menanti Bapak Ibu untuk dibantu memberikan riset-riset lanjutan untuk melengkapi data-data seperti misalnya potensi, kegunaannya, dan seterusnya. Ini juga ada data-data ya, status IUCN untuk jenis-jenis yang ada di kebun raya, di mana ini akan menunggu Bapak Ibu semuanya untuk dilakukan penelitian untuk upaya konservasi dan perbanyakan seperti itu. Dan untuk data-data yang lebih lanjut, Bapak Ibu bisa mengunjungi website makoyana.brin.go.id. Dan di sini ada data-data di jaringan kebun nasional, yaitu kebun raya daerah, dengan berbagai topik yang spesifik dari misalnya hutan hujan pamah Kalimantan, hutan kerangas Kalimantan, kemudian juga hutan rawa gambut Kalimantan. Ini Bapak Ibu bisa juga menggunakan jaringan ini untuk digunakan sebagai objek atau subjek riset dari para peneliti. Ini di sini di Makoyana Bapak juga bisa melihat bagaimana persebaran atau koleksi di masing-masing kebun raya. Dan ini di herbarium, Bapak Ibu ada di Kehati dan juga di gedung herbarium. Jadi kita memiliki ada apa namanya 3 gedung begitu ya untuk koleksi. Di sini selain yang ada di INACC, di herbarium dan juga di gedung zoologi, kita juga memiliki koleksi di gedung Kahati begitu yang di situ juga sebagai nanti diharapkan akan berkembang terus untuk mendapatkan koleksi-koleksi baru dari teman-teman. Nah, kemudian di sini koleksi ini juga dapat digunakan bersama-sama oleh terutama untuk program ini begitu ya. Dan ini juga ada koleksi dari zoologi. Memang koleksi kering kita masih simpan di MZB, di gedung MZB, kemudian koleksi basah sudah dipindahkan di Kehati. Teman-teman dapat nanti mengakses database-nya begitu ya. Kita akan mungkin jadi output pertama dari program ini untuk database kita beri nama Sekar begitu ya untuk database Kehati. Dan ini adalah lebih detail dari koleksi yang kita miliki. Kemudian ada mikroorganisme. Di sana ada sekitar 6.000 koleksi yang kita miliki dan insya Allah di 2 bulan ke depan akan ada tambahan koleksi repatriasi dari Jepang yang merupakan hasil kerjasama riset teman-teman senior kita di tahun 2005 hingga 2009 juga akan ditambahkan sebagai koleksi di INACC. Ini kalau adalah koleksi kayu, di Silarim ini masih data sebelumnya kita belum apa namanya, ini adalah koleksi baru di BRIN, koleksi kayu, bambu, rotan, fosil kayu, menghasilkan kayu seperti kulit dan seterusnya yang sudah kita tempatkan di gedung Kehati. Kemudian juga ada koleksi bank biji, sekitar 5 juta koleksi yang kita miliki, tidak hanya dari tumbuhan keras gitu ya, pohon, tapi juga nanti akan ditambahkan koleksi-koleksi dari teman-teman di OR pertanian dan pangan seperti koleksi padi, koleksi pisang, koleksi pakan herba dan pakan ternak dan seterusnya. Ini fasilitas yang dimiliki, kemudian sekitar 5 juta koleksi yang kita miliki, teman-teman bisa gunakan resource ini untuk riset teman-teman. Jadi ini adalah koleksi geodiversitas di Karangsambung, saat ini baru ada 500 koleksi batuan dan sedimen core yang ini saat ini juga sedang akan bertambah dengan koleksi dari ekspedisi marin bersama teman-teman Cina. Ini sudah masuk tapi sedang dalam pendataan di DPKI seperti itu. Itu ekspedisi laut dalam sudah dilakukan pekan lalu. Dan ini adalah koleksi arkeologi yang sudah ada di Kehati. Nanti untuk seluruh koleksi ini akan tempatkan di lantai 1, 2, 3 di gedung A di Kehati. Itu Bapak Ibu adalah koleksi-koleksi yang dapat digunakan, tapi kita tidak hanya terbatas pada koleksi yang sudah ada, kita akan dapat juga melakukan akuisisi data, melakukan analisis data lebih lanjut untuk spesimen-spesimen yang Bapak Ibu dapat dari hasil eksplorasi yang catatan tapi sudah diserahkan kepada DPKI untuk menjadi bagian koleksi. Nah, koleksi-koleksi baru tadi dapat juga digunakan untuk bahan riset Bapak Ibu semuanya, termasuk whole genome sequencing, setelah itu tercatat sebagai koleksi di BRIN. Dan Bapak Ibu, jadi untuk kegiatan ini gitu ya, untuk akuisisi data itu kita akan melakukan karakteristik dasar standar yang sudah teman-teman lakukan selama ini. Kemudian analisis data lanjutan ini sedang kami godok ya untuk topik-topik risetnya. Termasuk di sana analisis genetik dan fenotip Kehati, revisi taksa, filogenetik dan seterusnya untuk Kehati. Kemudian Tulang grup kita adalah kita melakukan peningkatan jumlah data koleksi hayati dan non-hayati, karena di sana data-datanya yang masih ada sekarang adalah data-data dasar. Seperti yang tadi saya sampaikan, itu adalah berapa jumlah koleksi, nama spesies, nama suku, nama famili, dan seterusnya. Kemudian di sini juga akan ada peningkatan kapasitas riset nasional melalui kerjasama dengan mitra global. Jadi kami harapkan teman-teman nanti dapat mengajukan proposal sesuai dengan topik yang kami sampaikan. Nanti akan kami sampaikan juga di website gitu ya, insya Allah dalam minggu ini. Itu Bapak Ibu dapat mengajak semua mitra global untuk bergabung. Termasuk di sana akan ada peningkatan kapasitas sumber daya riset kehati dengan menggunakan infrastruktur riset yang mutakhir yang terkini yang kita miliki di BRIN. Tujuannya adalah nanti kita juga akan membuat SOP, mekanisme penyimpanan dan mekanisme pemanfaatan, apakah terbuka atau tertutup ataukah terbatas begitu ya. Nanti akan kita jadikan output dari kegiatan ini. Nah, tetap aja luaran atau targetnya, meskipun ini di bawah Direktorat Pengembangan Koleksi Ilmiah, targetnya adalah karya tulis ilmiah internasional, materi internasional, kemudian ada karya intelektual dengan status minimal terdaftar, dan output lain yang disepakati ini adalah data untuk output lainnya. Kalau hasil ekspedisi akan menghasilkan spesimen baru, tapi untuk akuisisi data ini kita akan menghasilkan data-data baru, baik data yang tersimpan maupun database yang termanfaatkan. Ini sebelah kanan ke bawah adalah contoh apa namanya target begitu ya, penambahan data. Misalnya dari program akuisisi khusus untuk whole genome sequencing, baik dari mikroba, tumbuhan, hewan maupun mikrobiom ditambah human, nanti di mana tadi kita mensupport pembicara pertama dan pembicara kedua, supaya ini akan dilakukan juga di BRIN. Itu adalah contoh. Kemudian untuk siapkan infrastruktur, ini adalah alat-alat mutakhir yang ada di BRIN yang dapat digunakan oleh Bapak Ibu semuanya dengan mekanisme ELSA. Jika Bapak Ibu mendapatkan pendanaan dari program ini, Bapak Ibu akan mendapatkan top up ELSA untuk dapat menggunakan alat-alat untuk analisis lebih lanjut. Kemudian potensi tema dan subtema ini masih berkembang Bapak Ibu. Insya Allah akan kami finalkan dalam 1-2 hari ini gitu ya. Ini akan silakan Bapak Ibu bisa memberikan masukan baik terlangsung kepada saya maupun kepada kepala OR atau kepala PR masing-masing. Jadi untuk bidang hayati ini mungkin temanya misalkan analisis genetik dan fenotip, kemudian regresi taksa, filogenetik, whole genome sequencing untuk koleksi tumbuhan, hewan, dan mikroorganisme. Dan di bidang arkeologi ada penataan koleksi di 10 balai arkeologi, analisis dan fungsi koleksi gerabah misalnya. Kemudian untuk bidang kesehatan melakukan karakterisasi koleksi secara konvensional, molekuler, dan patogenesis, kemudian karakterisasi koleksi untuk bahan baku obat, DNA koleksi infeksius, dan seterusnya. Itu adalah tema-tema yang coba diangkat, yang teman-teman bisa masuk ke sana. Untuk bidang kebumian dan maritim, ada pengembangan koleksi jenis tanah di Indonesia, kemudian analisis koleksi batuan untuk komposisi mineral, petrografi, XRF, morfologi, dan compression test. Untuk koleksi sedimen ada ukuran butir dan morfologi, koleksi tanah ada uji ayak dan hidrometer, uji pirometer, dan seterusnya. Dan untuk bidang pertanian dan pangan, usulan dari teman-teman OR adalah melakukan integrasi atau data duplikat koleksi SDG, sementara genetik pertanian dari Kementerian Pertanian, kemudian pengembangan koleksi rejuvenasi dan karakteristik akses padi nasional, identifikasi ketahanan terhadap hama penyakit, identifikasi rekaman abiotik dan seterusnya, karakteristik 300 varietas dari International Institute, kemudian pengembangan koleksi hama penyakit di Indonesia, seperti itu. Ini adalah beberapa gambaran untuk data lanjutan yang akan dilakukan atau akan kita lengkapi untuk menyajikan data hayati dan non hayati Indonesia yang komprehensif. Jadi Bapak Ibu di sini bisa lihat ada 8 jenis koleksi yang nanti juga akan berkembang. Seperti yang saya sampaikan adalah koleksi dari teman-teman pertanian, koleksi tanah, dan seterusnya. Dari teman-teman kebumian ini akan terus bertambah, dan di situ akan ada data dasar yang kita sebut dengan SEKAR, database karena hayati Indonesia terintegrasi. Di sana hanya ada data seperti nomor koleksi, nama spesies, suku, asal lokasi, kolektor, tanggal koleksi, dan seterusnya. Kemudian dengan bantuan teman-teman periset semuanya, baik mahasiswa maupun periset aktif, gitu kita akan menambahkan data-data molekuler, data-data umur, periode, data-data kandungan mineral, analisis struktur menggunakan microcity, kemudian fungsi atau penggunaan, metode perbanyakan untuk koleksi-koleksi hidup, dan seterusnya. Ini masih akan terus berkembang, dan Bapak Ibu juga bisa memberikan topik-topik mungkin yang di luar ini yang kami anggap masih relevan. Dan ini khusus untuk webinar ini. Ini adalah program whole genome sequencing dan environmental DNA untuk Indonesian Biodiversity. Jadi kita akan menggunakan koleksi ilmiah yang ada di BRIN dari microorganisms, plants, animal, human environments untuk kita lakukan whole genome sequencing, baik itu de novo hingga kita menggunakan human whole genome sequencing yang nanti akan kita diskusikan lebih lanjut mekanismenya. Tujuan dari program ini di bawah akuisisi ini adalah untuk menambah data sekuensial data di RIN dan juga sebagai fondasi riset untuk pelestarian biodiversitas dan hutan berkelanjutan. Ini lebih detail untuk whole genome de novo sequencing untuk sampel baik hewan dan tanaman yang berupa sumber daya genetik endemik maupun yang terancam punah. Jadi ini adalah proses-proses yang dapat dibantu di lab sequencing dari preparasi sampel ekstraksi, kemudian lakukan sequencing hingga bioinformatika. Ini akan menambahkan data BGS di BRIN. Kemudian ini sampel yang dari mikrobial dari mikroorganisme yang ada di INACC. Kemudian sekuensi akan dibangun secara bioinformatika dengan referensi, berbeda dengan yang dua sebelumnya. Kemudian juga sampel yang berasal dari manusia Indonesia. Ini akan menjadi basis riset kesehatan untuk penyakit genetik, kanker, mekanisme patogenesis dan populasi manusia. Ini tidak akan tumpang tindih dengan program dari Kementerian Kesehatan seperti itu yang memang fokusnya pada penyakit. Dan kita akan ada environmental DNA. Bapak Ibu identifikasi spesies pada ekosistem seperti itu ya. Itu Bapak Ibu juga bisa lakukan di bawah program akuisisi data ini. Itu yang sekilas. Dan ini untuk mekanismenya saya sampaikan bahwa untuk pendanaan ini ada perubahan arahan dari Kepala BRIN, awalnya full seperti DIM, tapi akhirnya kita menggunakan mekanisme yang saling silang seperti itu. Jadi pendanaan untuk yang diajukan oleh teman-teman untuk program DIM implementasi strategis ini yang meliputi pembelian bahan pengujian, pembelian bahan seperti itu. Analisis data akan menggunakan ELSA point, di mana akan ditambahkan kepada teman-teman untuk melakukan analisis data. Kemudian akan ada mekanisme call for person untuk mahasiswa yang akan melakukan sekolah untuk menyelesaikan kuliah dengan target 16 mahasiswa per tahun. Kemudian juga call for research proposal untuk kelompok. Ini untuk detailnya akan kami segera sampaikan begitu ya di website pendanaan BRIN. Jadi Bapak Ibu akan banyak manfaat yang digunakan. Bapak Ibu akan dapat menggunakan koleksi di BRIN yang dimiliki oleh BRIN, dan kita juga nanti akan menggabungkan atau mengintegrasikan data dari seluruh koleksi yang ada di perguruan tinggi, dan kita berharap teman-teman dari perguruan tinggi juga akan terlibat untuk melengkapi data-data tersebut, di mana jika teman-teman dapat mengajukan proposal bersama dengan teman dari perguruan tinggi, kita juga dapat memanfaatkan dengan mekanisme yang akan ditetapkan koleksi-koleksi yang ada di luar BRIN seperti itu. Dan ini seperti Bapak Ibu yang mungkin sudah tahu di kegiatan di implementasi strategis lainnya, ini akan ada sub-sub judul riset yang akan mendukung kegiatan akuisisi ini, di mana nanti akan dikoordinasikan oleh pusat riset masing-masing gitu untuk pengajuan. Jadi Bapak Ibu tema-tema tadi juga akan bertambah sesuai dengan usul Bapak Ibu, tapi juga Bapak Ibu dapat berpatokan pada tema-tema yang sudah kita berikan. Nah, ini akan melakukan call for proposal untuk timeline mungkin setelah Lebaran akan dimulai. Kemudian akan ada seleksi administrasi dan seleksi substansi serta review RAB. Nanti akan ada panel untuk itu. Kemudian jika lolos seleksi akan ada penetapan SK penerima pendanaan dan penandatanganan kontrak. Selanjutnya untuk kriteria pengusul proposal untuk ketua minimal S3 dan anggota minimal S1, tidak sedang menempuh studi lanjut atau postdoc dan seterusnya, dapat berkolaborasi dengan institusi lain baik dalam maupun luar negeri. Kemudian teman-teman untuk yang persyaratan proposal ini sudah memiliki bukti pendaftaran atau pemilikan akun pada RIN. Karena nanti semua data itu akan di-upload ke sana, seperti itu Bapak Ibu. Dan ini untuk yang lebih detail nanti akan kami sampaikan di website. Demikian yang kami sampaikan untuk kegiatan ini. Silakan jika ada pertanyaan dan juga ada tambahan, terutama masukan-masukan topik-topik riset yang akan kita usung bersama-sama. Silakan teman-teman. Makasih Zahra.
Iya, terima kasih Bu Rati untuk paparannya yang sudah sangat jelas ya. Tapi mungkin Bapak Ibu ada yang memiliki pertanyaan langsung ke Bu Rati terkait platform kolaborasi riset baik yang hayati dan non-hayati ini. Karena sebentar lagi akan dibuka Bapak Ibu dan mungkin bisa disiapkan proposalnya. Mungkin ada pertanyaan terkait itu dari Bapak Ibu semua yang hadir di sini.
Atau kita bisa diskusi topik riset ya yang belum terjangkap, mumpung teman-teman semuanya sedang berkumpul.
Iya,
Saya bisa buka lagi ya, nggak apa-apa ya.
oh iya. Mbak Lina ada masukan mungkin Bu. Silahkan Mbak Lina disampaikan langsung.
Masukkan sih pertanyaan, Mbak Zahra.
Oh, ada pertanyaan? Iya.
Iya,
Nanti ini platformnya tadi Nanopore dan Illumina ya?
iya,
Apa
Kak.
kedua-duanya kan ya?
Iya.
Nah,
Untuk metodenya kita serahkan kepada teman-teman yang lebih paham.
oke. Yang berarti kan hanya whole genome sequencing. Kalau misalnya kita punya proposal dengan RNA-seq, transkriptomik, apakah akan diakomodir gitu?
Ya,
Karena
Mbak.
kan, karena kan whole genome sequencing itu hanya sequencing saja, tapi kita nggak tahu fungsinya apa. Untuk mengetahui fungsinya kan biasanya kita pakai transkriptom, karena kan dia di sini ada protein coding, di sini ada walaupun bisa secara ab initio, tapi lebih baik kan pakai datanya dari tanaman itu. Apakah memungkinkan ketika kita membuat proposal itu akan didanai jika tidak hanya genomnya, tapi dengan data anotasinya seperti itu.
Ya, udah bagus Mbak. Kita tambahkan ya Mbak Lina. Makasih ini masukannya. Ya, kita harapkan memang sampai seperti itu Bapak Ibu. Jadi tuntas untuk data yang dimiliki misal dari suatu tumbuhan. Kita catat untuk masukannya Mbak Lina.
Kalau tanamannya ini listnya apakah akan diberikan atau list dari tanaman yang kita teliti saja seperti itu, atau ada preferensi sepertinya tanaman ini seperti ada seleksi dari proposal itu.
Ya, jadi gini, kalau untuk spesies prioritas begitu ya, memang kami sedang diskusikan dengan kepala pusat riset dan ini yang lebih paham adalah teman-teman periset. Kalau misalnya teman-teman periset sedang memegang spesimen sedang diteliti, tapi itu misalnya akan didanai dengan kegiatan ini, teman-teman mungkin bisa serahkan dulu untuk wajib serah wajib simpan, kemudian teman-teman akan didanai untuk melakukan uji lebih lanjut. Nah, untuk mekanisme apakah spesimen tersebut dapat diakses orang atau tidak itu di RIN kita memiliki opsi untuk terbuka maupun tertutup. Jadi kalau misalnya teman sedang melakukan riset, kemudian sebelum ada publikasi, teman-teman akan menutup dulu atau menutup akses terhadap spesimen tersebut itu dapat dilakukan. Jadi untuk pertanyaan pertama tadi, list dari koleksi akan ada di database sekar. Itu juga sedang dipersiapkan oleh teman-teman dari Pusdatin. Tapi jika teman-teman memiliki spesies atau apakah tumbuhan atau hewan yang sedang dikerjakan dan akan dimasukkan sebagai penambah data dari ekosistem data ini, teman-teman bisa serahkan dulu melalui mekanisme WSWS. Jadi silakan. Jadi untuk prioritas spesies itu diserahkan sepenuhnya kepada teman-teman di OR karena teman-teman yang tahu mana yang berpotensi, mana yang sedang urgent misalnya gitu untuk harus diteliti dan seterusnya. Seperti itu. Nanti untuk reviewer juga dari teman-teman di OR seperti itu Mbak Lina.
Oke, terima kasih.
Terima kasih Bu Rati. Ini sepertinya ada Bu Asda yang ingin bertanya juga. Dipersilahkan Ibu Asda.
Ya, mungkin boleh disampaikan dari PR mana gitu ya teman-teman. Jadi
Oh
saya
iya.
bisa menambahkan di topik riset di bidang apa ya. Silakan Mbak Azda.
Ya. Terima kasih Mbak Sarah, Bu Rati. Sebelumnya saya mohon maaf ya Bu, saya tidak bisa ke kamera on-nya. Ini koneksi di KKB kami nggak begitu bagus ya. Nama saya Asta, saya dari peneliti dari Pusat Riset Perikanan Bu Rati. Jadi ORKM ya Bu. Uh, Pertanyaan saya hanya simpel aja. Tadi kalau melihat apa ya open call selanjutnya untuk kita semua, saya tadi tidak melihat ada komoditas-komoditas perikanan yang disebutkan oleh Ibu Rati. Ya, kebanyakan adalah komoditas terestrial ya Bu. Nah, jadi pertanyaan saya, interest saya sih saat ini itu adalah rumput laut Bu. Nah, apakah untuk call dari apa namanya kegiatan invitasi ini akan ada apa namanya komunitas-komunitas dari perikanan, Bu. Interest saya adalah rumput laut. Itu aja. Terima kasih.
Makasih, Bu. Sudah kita tambahkan ya di sini langsung apa namanya? Memang sempat juga ada usulan untuk ada penambahan koleksi ikan, tapi ini ikan yang hidup, bukan ikan yang jadi koleksi basah di zoologi. Jadi akan kami tambahkan juga untuk komoditas di teman-teman dari perikanan begitu. Nanti Mbak Lina bisa bantu tambahkan ya. Saya nggak begitu takut takut bahasa teknis, takut salah ketik. Bisa di chat, nanti ditambahkan di chat, saya langsung tambahkan di sini Mbak Lina.
Metatranskriptomik ya Mbak ya? Tadi itu
Iya.
metatranskriptomik.
Enggak, transkriptomik aja.
Iya, di chat aja deh. Aku takut salah nulis.
Oh ya,
Iya, silakan.
data transkriptomik.
Ada pertanyaan selanjutnya dari Bu Malamuria. Ya, silakan Ibu, dipersilakan.
Ya, Bu, terima kasih. Mohon maaf sebelumnya saya juga off kamera karena masalah internet, Bu. Ini dari penjelasan yang tadi Ibu Rati sampaikan. Oh maaf sebelumnya saya perkenalkan, nama saya Malamuria Ningrum. Saya dari OR Pangan dan Pertanian dari Pusat Riset Tanaman Perkebunan. Begini Bu, yang tadi sepertinya ada yang Ibu sampaikan kalau kita bisa melakukan duplikasi koleksi. Saat ini CWS saya di KKI Purwodadi, Bu. Nah, di KKI Purwodadi itu saya melihat ada koleksi agave. Nah, agave ini yang saya tahu bisa untuk salah satunya bisa digunakan sebagai bahan baku bioetanol. Nah, dulu saya waktu pas di Kementan itu kita juga punya koleksi, Bu. Di Balitas itu agave sisalana juga. Nah, jika memungkinkan bisa nggak kalau misalnya kita teliti lebih lanjut gitu, tapi mungkin harus ada penambahan koleksi yang dari Kementan gitu. Tadi makanya saya langsung eye catching dengan duplikasi. Apakah itu bisa memungkinkan Ibu kalau misalnya kita melakukan duplikasi koleksi yang dari Balitas? Karena gini Bu, namanya juga OR Pangan dan Pertanian ya Bu. Jadi memang di sana kan lebih utamanya koleksinya itu ke tanaman apa penelitiannya itu kan tanaman pangan. Jadi tanaman-tanaman serat sebenarnya agave itu bisa sebagai tanaman serat. Itu kurang mendapat perhatian gitu ke istilahnya karena kan ada tanaman-tanaman yang strategis. Nah, kalau misalnya bisa memungkinkan mungkin saya join dengan teman-teman yang dari KKI Purwodadi untuk pengungkapan potensi dari agave ini gitu. Jadi ada agave salana, agave macam-macam Bu gitu. Salah satunya sebenarnya dia kalau dulu utamanya untuk tanaman serat. Cuman saat ini bisa juga diungkapkan potensinya itu sebagai bioetanol. Mungkin itu Bu. Terima kasih sebelumnya.
Ya, makasih Mbak Mala. Langsung siapin Mbak Sarah ya.
Ya.
Jadi gitu Mbak Mala. Jadi memang saat ini dari teman-teman UL Pertanian Pangan kemarin sudah mengajukan list apa ya? Plasma nutfah list tetua juga gitu ya yang akan dilakukan duplikasi di sini di BRIN dari teman-teman di Kementerian Pertanian. Jadi kalau saya nggak cek satu-satu apakah agave sudah masuk atau tidak. Jika tidak ini teman-teman bisa mengajukan tadi melalui proposal. Kita akan pertama akan melakukan upaya misalnya melakukan pembelian kepada Kementerian Pertanian atau yang kedua kita akan melakukan kerjasama untuk memberikan duplikat itu dalam rangka juga wajib serah wajib simpan. Jadi agave tadi saya akan cek lagi kalau sudah masuk alhamdulillah, kalau belum berarti nanti akan bisa ditambahkan, bisa diusulkan Mbak Mala. Ini jadi teman-teman saya juga sampaikan nanti akan ada koleksi hidup begitu ya untuk kebutuhan riset. Jadi di sana ada koleksi istilahnya koleksi tetua gitu ya yang akan digunakan oleh teman-teman pertanian yang akan melakukan riset untuk tanaman pangan. Termasuk juga segala macam komoditas, duren, pisang, kemudian ada juga saya nggak hafal, tapi itu sudah sudah oke, sedang kita usulkan, sedang kita ajukan kepada Kementerian Pertanian untuk melakukan pembelian dari koleksi-koleksi yang ada di sana, seperti itu untuk dalam melakukan duplikat di sini. Jadi Mbak Mala itu memungkinkan insya Allah untuk kita menyatukan atau mengintegrasikan koleksi-koleksi sumber daya genetik yang ada di Indonesia seperti itu.
Baik,
Silakan
terima kasih
diajukan
Bu
nanti Pak.
Rati.
Iya,
Baik, terima kasih Bu Rati. Ya.
sama-sama Mbak Mala.
Terima kasih. Iya Bu Rati, ada juga dari Profesor Eni di chat mengusulkan bahwa mungkin bisa ditambahkan topik genotyping ya by sequencing untuk melihat sifat unggul tertentu tanaman mungkin nanti di bidang hayati.
Ya,
GBS.
saya
Jadi
tambahkan
tidak
Prof.
ada dari Prof Eni, jadi mungkin
Iya, saya tambahkan. Jadi ini memang usul-usul teman-teman semuanya. Tujuan kita tadi kan bagaimana kita mendapatkan informasi selengkap-lengkapnya begitu tentang satu spesies yang mungkin nanti akan jadi modal untuk bioprospeksi dan itu akan apapun yang teman-teman sampaikan ini kan kami tampung begitu ya. Karena nanti juga teman-teman yang akan melaksanakan dan tujuannya adalah untuk kita gunakan bersama-sama datanya. Silakan ini jika ada usulan bisa sampaikan juga langsung kepada saya. Misalnya nanti nomor kontak saya bisa saya sampaikan di sini. Ini mumpung kami sedang menggodok usulan-usulan topik yang akan dimasukkan dalam kegiatan ini. Gitu. Tadi ada lagi ya. Tadi yang Mbak Lina belum masuk ya?
yang Mbak Lina, ya Bu. Kemudian ini ada dari Ibu Pratiwi Prananingrum juga. Jadi beliau bekerja dengan lisen ya lumut kerak simbiosis antara alga dan bumi. Jadi harapannya adalah bisa mengerjakan metagenomik juga Bu. Jadi saya rasa mungkin nanti di bidang hayati bisa ditambahkan juga metagenomik dan metatranskriptomik Bu mungkin.
Metagenomik
Tidak
dan
hanya
oke. Tidak hanya
transkriptomik
whole
juga.
genome sequencing ya.
Iya, tidak hanya
Oke.
whole genome.
Ya. Ya, teman-teman silakan yang mungkin dari arkeologi juga nggak masuk karena ini ya. Tapi ya kan kami lempar lagi, kami kembalikan kepada teman-teman kampus begitu untuk menambahkan topik-topik yang bisa diakomodir dalam kegiatan akuisisi data ini. Teman-teman juga gini ya, jadi kita juga sebenarnya pengen melengkapi koleksi yang teman-teman nanti misalnya berminatnya tidak hanya pada uji lanjutan, tapi juga bagaimana melakukan review misalnya dari hasil-hasil riset yang sudah ada di perguruan tinggi untuk komoditas tertentu, untuk melengkapi database kita, kami juga membutuhkan hal itu. Jadi kami membutuhkan bantuan dari misalnya teman-teman mahasiswa untuk melengkapi database kita. Misalnya sudah pernah dilakukan riset terhadap spesies A, tapi belum masuk dalam database, kita pengen teman-teman membantu juga ke sana. Jadi kalau misalnya ada adik-adik mahasiswa yang sedang berminat untuk melakukan kegiatan kayak studi literatur kayak gitu, itu juga kita membutuhkan untuk menambah atau melengkapi database yang kita miliki gitu seperti itu. Yang punya adik kelas yang sedang akan mengajukan, jadi memang lebih banyak ke review, cuma nanti dia akan membantu juga jika tertarik untuk melakukan analisis pada yang lebih dalam pada suatu komoditas misalnya, itu juga memungkinkan. Jadi ini sebenarnya kita mengakomodir riset-riset yang kurang seksi sebenarnya teman-teman ya, yang mungkin sebenarnya dianggap sebagai riset dasar, tidak menghasilkan misalnya suatu produk, tapi karakterisasi gitu ya, itu kami coba akomodir pada kegiatan ini. Jadi memang nggak kompetitif mungkin kesannya, tapi memang kita butuhkan untuk melengkapi satu data Indonesia seperti itu. Ini kalau data ini tidak hanya tumbuhan ya Mbak Lina ya, tapi semua
Iya,
semua makhluk hidup bisa ya.
Iya, metagenomik juga sepertinya jangan difokuskan pada
Dari
lisian
Jen
saja,
ya,
Bu.
oke.
Saya rasa akan banyak.
Iya, betul. Metagenomik juga. Tadi sangat kiri.
Yang sudah masuk pada licin misalnya gitu ya.
Mungkin Bapak Ibu ada lagi yang ingin memberikan saran atau pertanyaan ke Ibu Rati terkait platform ini?
Nomor HP saya tulis kalau misalnya teman-teman nggak sempat mikir sekarang bisa saya tunggu mungkin sampai besok gitu ya.
Ini nanti ada sebaran dari PR juga tidak Bu? Atau nanti langsung ke Bu Rati?
Sebenarnya yang kami masukkan sebagai sebagai anggota tim gitu di sini itu ada kepala PR dan kepala OR gitu. Jadi kami berharap kepala PR itu dapat mengakomodir semua yang tim di dalam PR seperti itu, yang mungkin saat ini masih ada 5 OR yang kita identifikasi ya. Jadi kalau misalnya Bapak Ibu nanti atau teman-teman bisa mengajukan kepada kepala OR pada saat finalisasi proposal ini. Nah, apakah nanti kepala OR juga menyampaikan pada teman-teman? Mungkin saya coba rapatkan besok bagaimana mekanismenya. Tapi kalau misalnya teman-teman mau langsung kepada saya juga silakan disampaikan untuk saya tambahkan.
Baik, di chat juga ada Ibu Inenah Wirajana yang mengusulkan juga metagenomik tidak hanya pada lesion, tapi pada yang lain juga, tapi sudah kita tambahkan juga ya Bu.
Budaya alam
Kemudian,
Indonesia gitu ya.
iya. Kemudian dari Prof. Eni juga GBS-nya nanti bisa dipakai juga tidak hanya di OR Hayati dan Lingkungan, tapi juga bisa di pertanian dan pangan, bisa juga untuk tanaman liar dan cultivated. Jadi menurut saya nanti mungkin akan banyak yang pakai ya untuk genotyping by sequencing ini juga. Terima
Iya.
kasih Prof. Eni.
Makasih Prof. Ini dananya cukup besar teman-teman. Jadi ngabisin juga agak susah kayaknya kalau nggak dibantu. Soalnya untuk bahan aja. Tapi untuk pembelian tadi memungkinkan. Misalnya tadi Mbak Mala mau apa namanya melengkapi koleksi untuk agave gitu ya. Dan misalnya kita harus beli kepada pertanian, kita akan melakukan pembelian itu kayak gitu. Jadi kalau misalnya bisa belum masuk dalam mekanisme PNBP, kita bisa melakukan kerjasama itu sudah diajaki oleh Bu Puji untuk itunya, untuk apa namanya kerjasama dengan Kementan. Itu. Ya, silakan teman-teman. Ini mungkin nggak saya edarkan dulu, khawatir ini. Tapi teman-teman yang punya topik-topik yang akan diusulkan, silakan bisa langsung kontak saya dalam 2 hari ini. Saya besok insya Allah mau meeting dengan teman-teman di Kepala Pusat Riset untuk memfinalkan proposal ini gitu ya. Jadi selama itu mungkin 2 hari bisa terus bertambah untuk topik-topiknya. Gitu, Mbak Sarah. Silakan kalau ada lagi.
Iya, Bapak Ibu dipersilahkan kalau ada pertanyaan dan masukan langsung ke Bu Ratih. Mungkin terkait
Izin
riset.
Zahra,
Oh, silahkan Ibu.
ya. Bu Rati, salam kenal, saya Cici di PR Perkebunan nih. Eh, saya ingin bertanya tentang pemulihan presisi yang bibit unggul tadi di poin fokus-fokus risetnya. Apakah ini termasuk kepada tanaman budidaya ya, terutama untuk tanaman industri yang perkebunan seperti kopi, kakao, teh gitu, karet. Apakah itu juga termasuk? Lalu seingat saya buat tahun lalu itu kami pernah menyusun proposal untuk pemanfaatan apa ya untuk SDG tanaman perkebunan gitu. Jadi kita mengelis gitu tanaman-tanaman perkebunan yang kita punya untuk duplikasi. Apakah itu juga sudah bisa terakomodir di sini gitu ya di program ini seperti itu?
Itu kemarin dari Bu Winita ya,
Seperti
masuknya
itu.
ya, tepatnya Bu Winita Mbak Cici. Bukan Bu
Kami
Winita ya
Mas
kemarin.
hortikultura.
Iya, tempat Bu Winita bukan?
Kalau sekarang kami sudah pisahkan Bu ya, jadi ada
hortikultur sama
PR
perkebunan.
hortikultura dan PR perkebunan.
Oh ya, oke.
Ya,
Ya,
sekarang kami
saya
kalau
cek,
saya sudah di perkebunan gitu Bu.
Saya cek kemarin yang sudah diajukan oleh Bu Dinita untuk list tanaman-tanaman hortikultur. Nah, ini yang perkebunan sudah masuk atau belum? Saya harus lihat lagi. Itu karena kemarin sudah sekaligus pengajuannya gitu untuk dari teman-teman di ORPP untuk kita punya koleksi juga di sini. Kalau misalnya belum, nanti akan sampaikan kepada Pak Kapus ya, perkebunan ya. Itu
Iya,
untuk ditambahkan. Kalau kemarin
ya.
baru dari hortikultur yang mengajukan untuk duplikasi koleksi dari Kementan.
Iya Bu. Terima kasih Bu.
Ya, Mbak Ci, makasih.
Baik, dipersilakan Bapak Ibu jika ada masukan yang lain atau pertanyaan yang lain. Apakah sudah jelas semua ya?
Kalau masih berpikir, silakan nggak apa. Nanti ada waktu di kontak aja saya, nggak apa-apa.
Iya, bisa langsung dikontak ke nomor ini. Tadi ada yang masuk atau tidak ya? Saya yang salah. Baik, saya rasa sudah clear ya untuk Bapak Ibu semua terkait platform kolaborasi riset Hayati dan non-Hayati ini. Silakan dipersiapkan Bapak Ibu proposalnya, dan jika ada pertanyaan bisa langsung mengontak Bu Ratih. Ini sudah share number juga. Jika ada yang ada topik riset yang belum ditambahkan ya Bu, bisa langsung mengontak Bu Ratih. Jadi dalam waktu dekat akan ada open call for proposal ini, jadi silakan dipersiapkan Ibu, dan seperti yang disampaikan Bu Ratih, dananya cukup besar. Bagi yang tertarik dan juga tidak harus mengeluarkan dana analisis keluar, gitu silahkan dimasukkan proposalnya, nanti kirim invitasi strategis ya Bu ya di pendanaan riset kreatif. Ini ada pertanyaan lagi masuk Bu, terkait mahasiswa DBR apa bisa mengajukan proposal ya Bu? Tapi harus S3 tadi ya Bu kalau kirim invitasi strategis.
Ini maksudnya mengajukan proposal untuk riset atau untuk maksudnya untuk sekolahnya, untuk studinya.
Bagaimana siapa ini? Pak
David.
David Purwoko, apakah ini terkait proposal sekolah atau riset ya? Sepertinya
David berarti sudah sekolah ya, posisi sudah masuk ya,
untuk studi S3 Bu.
Sedang S3. Mas David.
sedang S3, oke. Yang ini akan saya coba saya tanyakan dulu ya, soalnya kemarin kita untuk mahasiswa baru sebenarnya, tapi kalau misalnya nanti apa namanya, jadi David ceritanya sudah masuk, kemudian untuk support S3-nya, saya pikir sih sebenarnya bisa ya. Tapi nanti maksudnya bukan ke call personal sebagai mahasiswa, tapi maksudnya call proposal. Coba diajukan aja, gitu.
Iya. Baik, selanjutnya ada dari Pak Dani Bu. Bagaimana dengan koleksi mutan? Apakah boleh diajukan juga untuk sequencing?
Ya,
Mutan.
pertanyaan bagus nih. Oke, saya simpen dulu. Mas Dani dari PR mana ya?
Regen.
Regen iya Bu, Regen.
Regen, oke. Regen sebenarnya masuk sebagai tim. Nanti akan saya diskusikan dengan apa namanya dengan Bu Ratih ya. Bu Ratih masuk sini. Jadi memang kalau untuk teman-teman di Regen kan mungkin bukan fokus pada misalnya beda sama teman-teman di PRBE begitu ya. Jadi untuk yang ini akan saya coba tanyakan dulu kepada saya diskusikan dengan Bu Ratih dulu ya, Mas Dani ya. Nanti kalau misalnya oke, nanti Bu Ratih juga akan bisa tambahkan untuk sebagai topik riset di situ.
Baik, ada lagi Bapak Ibu sebelum kita tutup? Mungkin yang ingin bertanya langsung pada Bu Ratih? Baik, saya rasa sudah selesai. Baik, terima kasih banyak Bu Ratih untuk paparannya. Alhamdulillah mudah-mudahan bisa memberikan gambaran yang jelas ya Bu, untuk Bapak Ibu semua. Mungkin ini juga untuk pertanyaan terkait lab sequencing juga bisa langsung ditanyakan via email ya Ibu Bapak Ibu. Ada email pusatsequencing@brin.go.id. Di sana bisa langsung menanyakan terkait sequencing ya, platformnya ataupun nanti riset yang bisa dilaksanakan di lab, dan platform apa saja yang ada di lab sequencing bisa ditanyakan via email. Jika ingin bertanya pada saya atau Mbak Lina, nanti bisa dicek di Intrabrin, alamat email kami, bisa langsung kontak ke email kami Bapak Ibu semua. Kemudian saya rasa sudah semua kita bahas, 3 pembicara sudah menyampaikan paparannya. So I will switch back to English because we have arrived at the end of our webinars today. And I would like to express my gratitude for all 3 amazing speakers. So Dr. Wibli, Dr. Chen Liao, and also Ibu Ratih, thank you very much for attending today's webinar. It is a very interesting talk, and also we have quite a lot of discussion and also fruitful discussion. So I'm happy with today's webinar. And moreover, the sequencing lab of BRIN will offer a variety of sequencing services. So we encourage the researcher in BRIN to stay updated and also stay tuned on the latest news of the lab sequencing lab in BRIN. So we will have another webinar. So please also join us in the next session of the webinar. Once again, thank you for the speakers, thank you for all the participants, and we are looking forward to see you in the next webinar. So thank you. I give it back to the MC, Ibu Loviana, Ibu Andalusi.
Okay, thank you, Dr. Zara, for leading a very interactive discussion. And thank you to all speakers for a very interesting presentation. Ladies and gentlemen, this is the end of our agenda for today. I'm Andalusi Arini. On behalf of the committee, I would like to say thank you very much to our speakers, moderator, and all participants. See you in the next event. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih ya.
Terima kasih Bapak Ibu semua.